My name is Elizabeth Elliott. I'm a paediatrician from Sydney, from the University of Sydney and the Children's Hospital at Westmead. And we've got 4 15-minute talks, and the first of those is from Professor Sharon Friel, who is a Professor of Health Equity at Australia's National University.
Let me see. Oh, thank you. Well, thank you. Delighted, really delighted to be part of the Academy. Thank you so much for having me. Thank you for nominating me. So I want to speak about some new work that is really just starting this year. This is work that's based on my ARC Laureate Fellowship, which is all about governance for planetary health equity. And so looking at the confluence of climate change, inequality, and health. And this builds on many years of work that I have done with Tony McMichael. Many of you would have known Tony, the late marvellous Tony McMichael, with Michael Marmot, with Fran Baum, and colleagues elsewhere across Australia and internationally, really trying to drill into this relationship between climate change, inequality and health. And of course, what we see is basically a planetary health inequity crisis, where the ability to lead a life with the equitable enjoyment of health in a stable ecosystem is being severely challenged. And the picture here on the screen is from Lismore. And really, all I want to make a comment about what was observed, what was happening with Lismore, was this intersection between climate change, profound social inequities, and then the consequences for health and health inequities. And Lismore has borne the brunt twice. So in 2022, the devastating floods, most of the population that experienced the devastating floods were among the most socially disadvantaged communities. And it was the same communities back in 2017 when the Northern Floods events happened, including where Lismore is located. The same communities bore most of the brunt then. And the key point is very little changed for those communities between 2017 and 2022 in terms of the underlying social conditions in which people live, which we know affects their health. And so a terrible, inadequate and inequitable adaptation to climate change was experienced. And so climate change exacerbates health inequities through the underlying social inequities. But I want us to think about the consumptogenic system. If we're going to do something about those climate-related health inequities, we've got to do something about the underlying drivers of both climate change, social inequities, and poor health. And I refer to that as the consumptogenic system. So this system of institutions, of policies, of business practices, of norms, of behaviours that absolutely incentivize and reward the harmful production and consumption of fossil fuel goods and services. And those same goods and services are often very bad for human health, and they're very unequally distributed and valued. And so if we say, okay, we want to do something about this relationship between climate change and health, we've got to do something about equitable adaptation to climate change, absolutely. We've got to do something about this consumptogenic system. This is the mitigation argument. But often the response, the policy response that we see is what's often referred to as the lifestyle drift. So eat more sustainable and healthy foods. Here's a leaflet to tell you how to do that. Turn off your lights. Go out there and buy an electric car because that's going to save the planet. Now, all of that is very important, but it is completely insufficient. We know it's very insufficient for responses to the health and the health inequities that we see in society, and it's certainly inadequate for climate change mitigation. And so the question that we're asking in the Planetary Health Equity Hothouse, which is the initiative that I've set up through the Laureate Fellowship, is why is there so little effective remedial action happening? And what are the conditions to help progress that positive agenda for change? And that means we need to think about these sorts of issues. It's a public policy issue, it's a government action issue. How can intersectoral public policy be good for the environment, be good for social equity, and be good for health outcomes? So optimising the possibilities of mitigation actions. Just think of the discourse and the discussions that's going on at the moment with the decarbonisation agenda. Imagine if we were thinking about the decarbonisation agenda through a health and a social equity lens. Then the question of what does market activity look like that is, yes, profitable, but is also achieving these planetary health equity goals and interests. And fundamentally, we do not have a fit-for-purpose governance system in place, otherwise we wouldn't be having all these problems. And so what's the reset? How do we get a reset in governance? And so just a little illustration of some of the issues that we're looking at based on some work that we've done in the past. So this is just to give you, if we think about the public policy side of things and how do we get coherence between different policies across sectors, there are so many interests at play in making and implementing public policy. This is just a little example of that mix at play within the food system, the food world. Excuse all the acronyms. There's at the global level, the Food and Agricultural Organization. I'm not going to go through them all. There's the World Bank, there's the World Trade Organization, there's the investors community, there's the Global Food Alliance organization, civil society. So a whole suite of different actors involved in trying to shape public policy. Some of them are concerned about trade, some of them are concerned about the environment, some of them are concerned about health, but not all of them are concerned about all of those things at all. And some of them are concerned about these particular interests over in the right-hand side, or these ideas. Relatively few are concerned about health, relatively few are concerned about health equity, relatively few are certainly concerned about equity more broadly. So there's a mix of actors, of interests, and of ideas that are always mixing about in the policy systems. And so what we're studying is how to elevate the planetary health equity, climate inequality, and health up in that agenda. And then in the world of commerce, we've been hearing about, so this is the commercial determinants of health field that various of us are involved in. Sandra De Maio mentioned the forthcoming Lancet series where we look at all of these different practices of the commercial sector. But I just want to highlight financial, because we don't often think about the financial sector when we think about health or certainly planetary health equity. You might have seen this slide. So this is, it's actually based in the US, but it's not a whole load different to what happens in other countries around the world, including Australia. So these are different companies, different manufacturers, different retailers that are in food manufacturers and retailers involved in the food system, determining what's available, what's affordable, what's physically accessible, what's desirable to consume. And we know the effects on health of some of these high fat, high salt, high sugar, junk foods. So this is the percentage ownership by private investors in those companies, in some of those companies. And I've just put a little red box around, so BlackRock, Vanguard, and State Street, private investor firms. They're the same private investors that have the largest shares in the fossil fuel industry. So remember, going back to the idea of having coherence between the interests and the goals of what happens in different sectors and different policy domains. When we think about these money managers behind the scenes influencing the decisions of these food companies and of the fossil fuel companies, it's the same people. There's a really important point of intervention to engage with those private investors if we want to do something about planetary health equity. And so just to wrap up really of the big question is, so how do you make that happen? How do you govern in a way that elevates planetary health equity interests, that elevates the ideas of health, of social equity, of climate change into these often contested areas? So we need a new narrative talking about the promise of planetary health equity, much greater state capacity to be progressive and proactive rather than reactive to these challenges, and then looking at who has influence. And the final slide is building on work that we did in our NHMRC-funded Centre for Research Excellence on the social determinants of health equity. Myself and Fran Baum led that, where we developed this health equity power framework, really trying to tease apart these different actors, their interests, their ideas, how they advance an agenda. We're going to do this for planetary health equity as well. So if you want to read any more about these sorts of issues, a shameless plug for a book that I did on climate change and the people's health, and our new Planetary Health Equity Hothouse website. So thank you very much.
Thanks very much, Sharon. We have a question down the front here.
Thanks, Sharon. That was fantastic.
The other thing that's happened now, of course, is Ukraine. That's really thrown a spanner in the works, hasn't it, in terms of all of a sudden, you know, the need for gas. Everybody's turning around and looking for lithium because of China. There's conflicts and, you know, the amount of money going now to arms across all the world, that's a real problem.
Yeah, I mean, I think what you're pointing to is both the specific issue of conflict and how that intersects with the whole sort of global supply chain across a whole load of issues. And so really the importance of thinking about all of this in systems terms. And I should have said that the methodologies that I use and that we're using within the hothouse work is drawing on political science, and drawing on systems methodologies to help us understand how some of these exogenous factors like conflict that then affect the supply of oil and so on, what does that mean when you put it into that very complex system of everything that I've been speaking about? What does it mean for planetary health equity ultimately? Yeah.
Thank you for that wonderful presentation. Ainsley Newsom from University of Sydney. You know, at this planetary level, my very lay understanding sort of is that it needs regulatory intervention or governance solutions that actually apply at that planetary level. Yet, of course, most regulation happens locally or within a particular jurisdiction and very rarely goes across international borders. Are you going to be considering kind of solutions that exist above that level in your work? And if so, I'd love to hear your ideas.
Absolutely. So with the governance reset, one of the things that we're examining is multi-level governance. So exactly that point of what happens and what can happen at the local, what happens, what can happen, what should happen at the global or the international level, and how are they interconnected. And there's a beautiful example in learning to be had from tobacco control. It's not the same, of course, as planetary health, but with tobacco control, we have the Framework Convention for Tobacco Control, the FCTC, an international legally binding treaty that has been enormously helpful for global health and global health equity, which really provides a legal framework in which national and subnational regulatory responses, policy and regulatory responses are operating. So the FCTC prevents governments like Australia from having tobacco company interference in their national policy deliberations and implementation. So absolutely examining the possibilities for something like that.
Thanks. Will you all join me in thanking Sharon for a lovely presentation? Now, our next speaker is Professor Ian Freckelton, a Professor of Law at the University of Melbourne. Welcome.
Thank you, and good morning. Can I acknowledge the Arrente and Warlpiri people on whose lands I'm currently residing for some months, as well as the Wurundjeri people who are hosting us now. It's an enormous honour to become a fellow of this academy, and I hope to say some things about how I aspire to be able to assist the activities of the academy. I lead a mixed, perhaps a hybrid life, and that has its advantages and its disadvantages. I'm one of those creatures now known as a King's Counsel. I took silk 15 years ago, so I live and play arguments in the courts. More about that in due course. Been a member of a number of disciplinary tribunals. I'm a judge of a Supreme Court in another country. And I've been an academic in a variety of different disciplines over the years, most particularly law and also psychiatry. I have a lifelong interest in interdisciplinarity, the ways in which different disciplines can be drawn upon to mutual advantage, and part of that's involved my creating and editing the Journal of Law and Medicine and the Journal of Psychiatry, Psychology and Law for the last 3 decades. But perhaps it goes further than that. Much of the other activity in which I've been engaged has been to do with how the law, at a very practical level, utilises the fruits of the health disciplines and the scientific disciplines. And so I started in days when I worked with Michael Kirby in constructing a new evidence code for Australia, reflecting on expert evidence, and have gone on to write a variety of books on the subject, one of which is just going into its 7th edition. So it's a practical level of the application of interdisciplinarity. Another aspect in which that's been a part of my life has been working on mental health law. I've sat on the Mental Health Tribunal of Victoria for longer than I want to remember now, and was president of the Australian and New Zealand Association of Psychiatry, Psychology and Law for a significant period. So again, teaching about, working in, and reflecting about the intersection between law and the health sciences. In more recent times, I've been doing quite a deal of work to do with COVID-19 and the law and pandemics more broadly, and public health law, thinking about, to pick up what was just being spoken about, issues to do with One Health and its approach to how we can meet the challenges of COVID-25 and zoonotic diseases more generally. And just starting to think through those sorts of issues with how we handle monkeypox, the latest public health emergency of international concern. There's so much to be done in respect of how we grapple at a regulatory and practical level with challenges coming from zoonotic provenances, how we can respond quickly, humanely, in a way which respects rights, but which is effective in inhibiting disease transmission. So those have been things where I've worked particularly in the intersections, but also in relation to regulation of health practitioners in particular, as a member of tribunals that have had responsibility for doing just that, but also looking at where misconduct in the scholarly domain can distort information transmission. And we've seen that just latterly in respect of COVID-19. We've talked about hydroxychloroquine just recently, but there have been a number of scandals which have encompassed the New England Journal of Medicine, the Lancet, and others, where material that was expedited in publication has, as it turns out, not just been short of the mark, but been utterly fabricated and sinister in its ramifications. And those areas of scholarly misconduct have been a long-term interest of mine, and I think are important in terms of how not just we all conduct ourselves, but the environment that we construct and the guidance and mentoring that we provide to others who are emerging in our professions. I wanted to finish up with two matters with you, which might be a little different from some of what's spoken about in the academy. The first is a particular area, and then I wanted to offer a few observations about the interface between law and the health and medical sciences. I have found myself, through no particular aspiration, engaged in a variety of the most notorious inquests in Australian history in recent times. It started with a series of fatal police shooting inquests in Victoria. We had another one in relation to 9 very sad deaths in Kew Cottages, which was an institution for those with intellectual disabilities. Kumanjai Briscoe in the Northern Territory was a vital inquest which changed the way in which people are looked after in custody when they've been brought in intoxicated into police cells. The David Wilson inquest involved an Australian who was killed by the Khmer Rouge, and where an inquest was held many years later into his death to try and understand better how Australia attempted to intervene and negotiate his release. The Lent Cafe inquest in Sydney, little need be said about. The Bourke Street Mall inquest will ring many bells for people in recent times. I thought I'd just share with you a couple of comments about the Kumanjai Walker inquest, which is what leads me to Alice Springs for many months at the moment. These inquests are vital parts of how we learn. And again, it's been part of my commitment as a practitioner across disciplines to work through those ramifications. The death investigation book, which I wrote with Professor David Ranson, a forensic pathologist, is, I suppose, the main scholarly work that's utilised in Australasia in relation to inquest law and practice. But the area of coronial investigation is just fundamental to how we learn about avoiding avoidable deaths. And the Kumanjai Walker case is an archetypal example. This was a 19-year-old man who appears to have had FASD, had a long history of criminal offending in spite of his early age. Police intervened to arrest him, but they came from the outside, resonating all sorts of issues to do with the Coniston massacre in 1928. They came with long arms and they intervened in a way which was ill-considered, culturally insensitive, and which led to tragic consequences. The great thing about the coronial area is that it affords an opportunity to learn and to do better and differently. And what one can see multiple weeks now into this inquest is an attempt by a range of government and other instrumentalities to learn those lessons. And for a large number of senior barristers and solicitors at the bar table, vast numbers of witnesses giving evidence, but it is an inquest that is going to change the life of Australia and how we respect and think about racism at both a subtle and an overt level. And it's going to change how people are brought into custody, and it's going to look at the provision of healthcare in remote communities. It's exciting. It's grueling and difficult and challenging in all manner of ways, but it brings together medicine and law in a really constructive way. So I'd like to finish with you by just offering one or two observations about the relationship between law and medicine, which is where I hope that I can facilitate and provide some value to the Academy. There are all too many lawyers who give the law a bad name, who are obstructive, who are narrow, who are rigid, who are rule-focused. But there are other ways of utilising the law. And those are ways which assist and enable the medical and health and science areas to function effectively. They ask questions, they challenge, but they guide practice in ways which are thought-provoking and which are sensitive to environments and social changes. That's what I am hugely committed to and what I hope to be able to assist the Academy with. There was an area which was developed in the early 1990s called therapeutic jurisprudence, which many of you will have heard about. And it was generated by two United States academics who identified the fact that law can be counter-therapeutic in so many ways. But they tried to look at the other side of the coin too, namely how law can contribute in a pro-therapeutic way in how it is written, how it is interpreted, and how it is applied. It requires a bit of a shift in perspectives to reflect upon that potential role for the law, but it's changed a good deal of thinking and practice. And I think if one brings that kind of lens to bear on how the law can work with and facilitate research and practice within health sciences, that opens up exciting potentials. And that's what I'm hoping to work with you in doing. Thank you.
Have we got any questions for Ian? Ian, you mentioned that this child had fetal alcohol spectrum disorder, and we've heard Banksia Hill detention centres in the news again this week, where we know 36% of children at least have severe cognitive impairment. How can medicine and law work better to perhaps prevent some of these children who have health problems rather than justice problems ending up in the justice system?
It's a big and incredibly important and difficult question. We're only scratching the surface, I think, in really understanding how FASD is contributing to socially problematic behaviours right around Australia. It's not just an Indigenous issue, but it's massively overrepresented in the Indigenous population across a number of our states and territories. The straightforward answer is that we need early identification and intervention, and then guidance and support, because otherwise that trajectory into criminal offending and then into harsh responses by the criminal justice system will take place. But there are other ways, and the Supreme Court in Western Australia has been crying out for that through its Chief Justice. There's an increasing awareness throughout the law of these problems, but we're only inching towards solutions in them. But a start is identifying them, acknowledging them, and the need for change.
And of course, their origin is all in the social determinants of health. Have we got any questions from the audience? Yes.
Russell Gruen from ANU. Ian, that was amazing. Thank you for that presentation. I must say the tone of the coronial inquest is vastly different to the legal trial that is completed,
Yes.
and the taste that I think that left in everybody's mouth. How do those things go together?
Well, they go together in that they have completely different agendas. In a criminal trial, we try to work out whether the prosecution can prove beyond reasonable doubt that a serious crime has been committed, in this case, whether a police officer, by using excessive force, had murdered the victim. And that's a binary kind of answer, yes or no. He got the benefit of the doubt ultimately from the jury. But a coronial inquest is an inquisitorial inquiry into the circumstances of death, directed toward learning what we can identify as things in respect of which we can do better. Where did things go wrong? What were the precipitants in a broad sense? So it doesn't focus on criminal culpability, it's looking at systems issues. And that's where there's the real potential for constructive change.
Thanks. Another question?
Sue Fletcher from Western Australia. We know that repeated trauma damages a young brain. It alters the size and the structure of the brain. It can do the same to adults.
Yes.
This is something that's not going to be easy to diagnose. So we know through the work of the wonderful Helen Mulroy that this is real. The data is there.
Yes.
It's going to be very hard to convince courts or the police or anyone else that this child has a problem because they are damaged when it's something that is so difficult to diagnose.
Yes, you're raising an issue about trauma generally, and how trauma contributes to problematic behaviours and how we can incorporate trauma-aware responses, not just by health service providers, but by police and the legal system, is a huge challenge. And I'm glad you mentioned it because it's becoming an increasing focus in the Kumanjai Walker inquest. Like so many communities, Yuendamu has been afflicted by generational trauma. It doesn't date back to a 1928 Coniston massacre, but the state-sanctioned killing of so many people as a retribution certainly was a terrible contribution to the trauma experienced by persons coming out of the Tanami Desert. But in so many of these communities, there's been serial and repeated evolving trauma which has impacted upon people from one generation to the next and which can be re-triggered so readily. So it's a complex issue in environments where there can be a lot of violence, a lot of substance abuse, but also a background which is traumatic in so many disparate ways and which goes back generations. So it's certainly something that we have to work on in terms of not just identification of the physical realities and evaluation of the psychiatric sequelae, but how we as a broader Australian community can respond to it.
Thanks, Ian.
Thank you.
Thank you very much, Ian. And now I'd like to welcome Professor Susan Fletcher, who's the Chief Scientific Officer for PYC Therapeutics Limited.
Thank you. So I hold positions at PYC Therapeutics and at Murdoch University, and company policy says I have to tell you that anything I say is my own view and not the company view. I have spent my entire academic career working on rare diseases. I'm going to briefly mention two of them today. Rare diseases affect 7% of the global population, more people than cancer. Duchenne muscular dystrophy I worked on for the better part of 25 years, and our lab delivered 3 therapeutics which have gone through FDA approval in the US and are now in the clinic. Our work on inherited blindness, inherited retinal diseases is in the preclinical stage. So when we started working on Duchenne, we were really at the beginning of what we like to call the era of biopharmacotherapy. Conventional drugs target proteins. About 1.5% of our genome codes for proteins, and only a small number, 10-14% of those have druggable targets. Recombinant DNA technology brought us the enzyme replacement therapies. We used to jokingly refer to the costs as genzyme units because they're very, very expensive drugs. They're hard to manufacture. Proteins have to be folded, modified. They have stability issues. They're large. They're hard to deliver, and many people will develop inhibitors. So we, in the early 90s, we sort of entered the beginning of the RNA therapeutics age, and this allows us to target the source. If we know the gene, we know the problem, we know the mechanism, and it gives us a target that we can use the ultimate precision therapeutics, which are the RNA medicines or RNA targeting medicines to address these diseases. And monogenic and rare diseases really present opportunities that you don't see elsewhere, largely because of a regulatory environment that allows reimbursement
that encourages development of these therapeutics. But these are the ultimate precision medicines. So if we have two healthy copies of a gene, or on the X chromosome, one healthy copy of the gene, we have a healthy, normal functioning protein and a healthy individual. A mutation on either one or both copies of the gene can give us an abnormal protein, no protein, or an abnormal RNA, a regulatory RNA, and a patient with monogenic disease. This can be dominant negative gain of function, as we see with many of the MND-causing mutations, the small number that are familial. Recessive X-linked haploinsufficiency presents particular opportunities. And then we have, some of them are characterized by incomplete penetrance, which gives us a disease threshold, a likely disease threshold, and then the nasty repeat expansion diseases. So just looking very simply at gene expression, we have a DNA gives us a messenger RNA, but this is not fit for purpose. The RNA that comes off the gene is not fit for purpose. It has to be processed, it has to be spliced and modified to be useful as a messenger RNA or a regulatory RNA, and then that will give us a protein. So I'll go back to the RNA. The RNA is actually produced as a much larger molecule. It has untranslated regions that don't contribute to protein expression, another one here at the 3' end, and it has exons that carry the protein coding information interspersed with introns, which used to be called junk DNA, but really are very, very useful things. Take my word for it. So this is then spliced, and we can join these exons together in different ways to give us multiple messenger RNAs and multiple proteins from one gene. This is why we can have the same number of genes as C. elegans, which is a tiny little fat worm, tiny little worm about 1 centimeter long. We have a genome many, many times the size. We have the same number of genes, and we are a very different organism because we use those genes differently. This gives us another opportunity for intervention. So I worked on Duchenne muscular dystrophy, as I say, from the early 90s. We looked at Duchenne and the milder allelic disorder, Becker muscular dystrophy, and the wonderful Monaco lab, Lou Kunkel, Tony Monaco, determined the difference between Duchenne and Becker. Duchenne patients are in a wheelchair by 12. In the 80s, they succumbed usually in the mid-teenage years usually from respiratory infection and or cardiac failure. Becker muscular dystrophy patients can have an almost normal lifespan. They can have really good function. They're still at risk of cardiomyopathy. They do still have a problem. But the difference is the mutations in their dystrophin gene, the gene responsible for this disease, allow production of a protein with at least partial function, whereas in Duchenne patients, the protein, there is no functional or almost no functional protein. Duchenne affects all muscles, including the heart, and is invariably fatal. So if we have a look at muscle, this is a muscle section from a healthy individual, this is from a Becker muscular dystrophy patient, and this is from a Duchenne patient. And this muscle, it hardly represents muscle. These sections are stained with an antibody to identify dystrophin. In the protein cartoon below, the actin-binding domain of the protein binds to the cytoskeletal actin, and the C-terminal region binds in through a group of proteins called the dystrophin-associated complex, links through to the extracellular matrix, which in muscle is the basal lamina. And it's this link that gives strength and stability to the muscle during contraction. In the absence of this, the muscle fibers are fragile, they break down, the muscle regenerates, and we have cycles of degeneration, regeneration, and mononuclear infiltrate, all the inflammation that goes with the damaged muscle and the laying down of scar tissue. So if we have a deletion in this middle region, which is it's like the coils of a spring, it's a repeated region, a deletion there that doesn't disrupt the reading frame, doesn't disrupt the way the message is read, the patients will have Becker. Whereas if the mutation takes off the end, the C-terminal end that binds through the extracellular matrix, we have a protein that's anchored at one end, if it's made at all, and floating around and unable to function. This is an image of the gene structure. It's the largest gene we have. The average gene is 30,000 bases. This is 2.5 million bases. So it's not a normal looking gene. And these boxes and chevron side shapes are the exons and representing whether they, if they're removed, they disrupt the message or don't disrupt the message. So if they've got red sides and they're a box, you can take them out And you can still have a partially functional protein as long as you don't remove crucial functional domains. So a patient who has a deletion of exon 50, which is relatively common, will have all the rest of his information there, but it can't be read because the message is disrupted and he will have Duchenne. A patient who has a deletion of exon 50 and 51 has Becker muscular dystrophy. He's missing a hinge. It's not a perfect protein, but it's a whole lot better than having no protein at all. So on the 19th of September in 2016, this work, we first got proof of concept in 1996. So you can see it's a 20-year journey. During this time, half the patients who were alive when we started this work were no longer with us. That's the harsh reality of working on rare diseases. So this is a genetic drug to restore dystrophin expression by skipping one of those exons, skipping exon 51, to rephase the gene message. Whether it extends life, we don't know, but it certainly has shown some benefit in some patients. It's a lifelong treatment. It's an enormous impact on patients and their families. These kids miss a day of school every week. It's a weekly intravenous infusion. They have a port that allows them to do that. So this is just showing this part of the gene again, the gene message again. This is a deletion of exon 50. Nothing can be used. This is a BMD patient, and patients with this mutation have between 3 and 100% of healthy levels of an almost functional protein. Eteplirsen, which was then renamed exon dys 51, skips exon 51, and this is a result.
I can't. See, I... Why'd you...
So it's not a cure. He still has muscle disease. He's still weak. He's still going to end up in a wheelchair one day. But for a period in his young life, he will maintain some independence. And for young men who have this disease, just when other kids are out there pushing the boundaries, thinking about girls, causing havoc, thinking about driving, these boys are dependent usually on their mothers for everything, including toileting. And we had one young man who his mum sent us a video, and it was, "Look, I can pee by myself." So for him, that was a life-changing moment. I call this our interactome. So these are some of the people and the organizations involved in supporting us and working with us through this very long journey. I particularly acknowledge Muscular Dystrophy Western Australia, who funded us in our program in its entirety for the first 5 years when we could not get NHMRC funding. In fact, we got an NIH grant for this work that we couldn't get NHMRC funding for. So we got a $1.25 million NIH grant, which was really very important. I'll very briefly touch on the other the other programs. So this is work I'm doing with PYC Therapeutics, but it came out of a PhD project at Murdoch University, where I hold my academic position. So inherited retinal disease cause blindness. Some are more slowly progressive than others. We are working on 2 diseases, retinitis pigmentosa type 11 and autosomal dominant optic atrophy. They have different symptoms. The diseases progress differently, but really, most children have some symptoms of visual disturbance before they're 10 years of age. In RP11 and most retinitis pigmentosas, it's loss of night vision. Some of them never have night vision, so they don't know what it's like to be able to see in dim light. Restricted field of vision, loss of visual acuity, and blindness. So the reason I started working with PYC Therapeutics is we thought we had some nice molecules, but they're very inefficiently taken up by cells. So the morpholino oligos, which are the ones that we use, really do not get into cells very well. PYC has a diverse library of peptides derived from all archaeobacteria and viruses. This is that group there. So there's a proprietary way of fragmenting these libraries and identifying cell-penetrating peptides. And just for reference, this is all humans, all animals, plants, and fungi. We put one of these peptides onto our oligo, and we can deliver it into cells. We can, looking at retinal disease, loss of photoreceptors, death of the RPE, and by altering genes and altering the way genes are expressed, we can overcome some of these problems. And just here, these are approved nucleic acid drugs. It's not there for you to read, it's just for you to see there's a long list. These account for about $3.5 billion of spend or return to the biopharma sector in 2021. these are the drugs that are going through various stages of clinical development, and the spend is expected to be $8 billion by 2025. Regulatory agencies, government agencies, and health insurance agencies need to be ready for this this burden on the cost of healthcare coming forward if we are going to continue to treat rare diseases. And in Australia, we could do it a lot better. We have academia and clinical research sitting alongside various philanthropic organizations, federal funding bodies that are all kind of, it's a bit bitty, a bit disconnected, industry sitting off the side. This is how they do it in the US. They work much more closely, and nearly every biopharma company is in bed with a good number of academic labs. And we need to divert money from really speculative things like mining to equally speculative things like biotech drug development. Thank you.
Right, we've got time for some questions. Anyone got? Susan, this is a very slow process. Do you think that in the last 20 years, there are techniques that are going to enable these drugs to be more rapidly developed?
It's been enormously helpful, the RNA sequencing technologies. When we started this, non-coding RNA didn't exist. We didn't know how splicing worked. We just gave it a try. We had a budget that paid for 3 oligos. One of them worked. If those hadn't worked, that was it. We didn't have more money for it. So it's the technology, both the sequencing technologies, the RNA technologies, the manufacturing of these compounds, and these are, we use synthetic oligonucleotide analogs. So that has been hugely, hugely helpful. The high throughput screening and sequencing has really shortened the timeline. People now expect it to be a lot quicker. And sorry, just before it's very remiss of me to have not thanked the Academy for this incredible honour and the privilege. I was so concerned about going over time, I forgot to thank the Academy, but I really feel incredibly honoured, and I'm so glad to be in the company of so many giants that I admire.
Well, I think we all feel privileged to have such an exciting group of new fellows. And the final presentation for today is Professor Denise Doolan, who's a Professorial Research Fellow in Infectious Disease Immunology at James Cook University.
So thank you. I'd first like to acknowledge the Wurundjeri people of the Kulin Nation on whose land that we meet today, and pay my respects to the ancestors and descendants. And I would also like to thank the Academy for welcoming me as a fellow. It is an enormous privilege, and I am really delighted to be present here. And I hope that what I'm going to do today is to present some of the little bit of snippets of some of the research that I've been doing to sort of give an indication of how I think that we might be able to contribute to the Academy. So my work is predominantly looking at systems biology, so integrating systems biology and immunology to try and design vaccines, diagnostics and therapeutics. And so a lot of the work that's already been presented today has been talking about all the different omics. And what we're really trying to do is integrate sort of particularly the transcriptomics, proteomics, and immunomics, where the human immune system interacts, and try and integrate that and develop computational pipelines validated to come up with ideally new vaccines, drugs and diagnostics. And so I like to sort of think of the work that we're doing with this cartoon, which you've probably all seen, and really what we're doing now is taking a holistic approach to understanding how to design new vaccines, drugs and immunotherapeutics. So you'd sort of argue that traditionally science has been a reductionist science where things have been taken apart. But what we're really doing now is trying to understand on a whole and understand how everything is put together. And a really important part of this is cross-disciplinary science. So trying to make use of all of the expertise from different disciplines. So the focus of my work has really been on chronic and infectious diseases. And what I'm showing here is the global burden of disease by cause and by country. And what I really want to highlight is that the chronic infectious diseases are highlight very high on these lists, and a lot of them map to the tropics. And it's important to note that about 50% of the world's population will live in the tropics in the next 30 years. So much of my work has been done with malaria as a prototypic chronic infectious disease. Malaria has been arguably around for a millennium, and it's been claimed that it has caused the death of half the populations, half the people that have ever lived. Might be an over-exaggeration, but nonetheless, it certainly is a very, very globally prevalent disease. So it has been an extreme challenge because it is a complex multi-stage parasite. It has co-evolved with the human host, meaning that it has evolved means to evade the host immune system. But really, we know very little about the mechanisms of protective immunity, and we know very little about the antigenic targets of protection, so the targets that we need to include in vaccines. So the parasite expresses 5,000 proteins. Which of those do we need to include in a vaccine? So that sort of that provides a foundation for the research that I'm doing. And really, we've been looking in 3 different areas. One is rational genome-based vaccine design. Another is trying to understand host immunity. And then thirdly, looking at biomarkers of disease. So I just wanted to give a couple of snippets of some of the work in this area. So with regard to vaccine design, as I mentioned, we're really trying to identify from the whole genome the small portions of the proteins that are important for induction of protective immunity, and that can be included in a vaccine. So in the case of malaria, which of the 5,000 proteins are the most important for including in vaccine? So a fair bit of my career has been trying to develop platform technologies to handle this challenge. And one of the platforms that we developed was in conjunction with Phil Felgner at the University of California, Irvine, and its protein microarray platform. And really from this, what we're able to do is to take from the complete proteome, identify the open reading frames and encode them into a expression vector, express them in a high throughput manner, and then put them onto a microscope slide, which can then be probed. And really what you get is you can put onto a microscope slide either a whole genome, so 5,000 proteins or 200 proteins, whatever, and you can then get this series of antibody reactivity, and you can screen samples of serum or plasma from individuals naturally exposed to a disease or experimentally infected. And basically, each of these proteins is expressed down here, and what you get is red are the proteins that are recognised, greens are the proteins that are not. So this technology gave rise to a company, Antigen Discovery Inc. in the United States, and it's been applied to 55 different infectious diseases, identifying vaccine targets and diagnostic markers, and has been recently implemented as a core platform in the Bill and Melinda Gates Global Health Vaccine Accelerator Program. But in the context of malaria, what we've done is we've been able to apply this platform and shown that of the entire proteome, only about 30% of the proteins are recognised by individuals who are immune, so that could be good candidates for vaccines. And also, importantly, we've shown that a lot of these novel antigens are much, much better candidates than the ones that have historically been identified. So the other approach that we did was looking at T-cell screening. So we're looking at malaria, an intracellular parasite, and many of the diseases that are challenging to develop vaccines require the induction of T-cells. And trying to identify which parts of the proteome are good targets of T-cells has been a challenge. So working with Alex Sede and colleagues at EpiImmune and now at La Jolla Institute, we actually took advantage of the fact that the computerized algorithms could identify parts of the protein that were highly recognized by T-cell. So predict the CD4 and CD8 T-cell epitopes within the algorithm. And we showed proof of concept for the T-cell epitope prediction for the EpiImmune algorithms, which gave rise to this immune epitope database, which is now widely used by researchers with infectious diseases, chronic diseases, allergies, cancer, biowarfare agents, for all T-cell type things. So what Alex and I thought is that we could actually use these T-cell epitope predictions in reverse. So really trying to apply them to the whole proteome to try and identify which of the whole proteome would be good candidates for vaccines or diagnostics. And when we applied this to malaria, again, what we have presenting here is a heat map where we identify some of these antigens from the proteome which are highly recognised and some of them are not. And again, we have found about 30% of the proteome is recognised by reproducibly recognised, but only a very small portion are really good targets. So since we had these proteome-wide data sets, we then wanted to look at the intersection between them and see if we could find anything that was useful for implications of vaccine development. And indeed, what we were able to find is that, contrary to what was expected, that we actually found that there were very distinct clusters of antigens recognised by T cells or by antibodies. So you're looking at T cells or antibodies here, and it's very clear that there's clear separations with the PCA plots. And what this really means is that distinct vaccine approaches are required and different antibody targets are required depending on what sort of vaccine that you want to use. So if you want to induce an effective T-cell vaccine against an intracellular pathogen, then you really need to actually rationally design a vaccine to do that. And interestingly, we're also able to use a support vector machine classifier to predict the antibodies and T-cell targets. And then we went and applied these and we identified some novel antigens and we assessed the ability of whether these antigens could protect in mouse models of malaria. And we used two schemes. We were able to use homologous, so immunising with the Plasmodium uelii mouse versions and showing that some of these antigens were actually able to reduce parasite burden in the liver stage and blood stage. But even more importantly, we were actually able to show that we had the human malaria, falciparum. If we express these and we immunise them in mice, that we're actually able to get also some reduction of liver stage and blood stage parasite burden. And the importance of this is that the Mosquito X vaccine, so the RTSS vaccine that has been given perdition, that's currently being rolled out by GSK, has absolutely no cross-species protection. And so if we're trying to develop a vaccine against a parasites such as malaria, where there are multiple strains, we really want to identify cross-species protection. So I want to just briefly switch to host pathogen immunity. And again, what we're looking at here is looking at systems immunology to provide an unbiased assessment of immune systems and interactions. So again, looking at all the different omics and computational approaches, and then ultimately trying to use these to look at biomarkers of signatures. And a particular interest in this space is trying to understand immune variation in individuals, because it's well known that the clinical outcomes and responses to infections are diverse. And we have this spectrum which ranges from complete asymptomatic to fatal. And this can be seen with sort of parasites, viruses, bacteria, and also particularly recently with SARS. And it's also known that with vaccination, you also get a variation in response where some people have successful vaccine take and some have complete failure. So what we're interested in is trying to understand the molecular basis of this. And in doing so, what we're doing is we're taking use of a controlled human infection model of malaria, which is developed by James McCarthy and Michael Good at QAMR. And malaria-naive people can be infected experimentally with parasites, and then they get this variation because they're naive, the parasites grow in their blood. But you get this variation in parasite growth rate between individuals. And because we've got a model where we have the pathogen factors and the vector factors constant, we can understand host immunity. And what we're able to show is that individuals were actually able to segregate into different areas. For instance, some individuals were actually able to have high microRNA responses, and these associated with better control of parasite burden. And more recently, we've expanded this, and we've actually been able to show and identify microRNAs and mRNAs, which actually correlate either positively or negatively with parasite growth rate. And so this allows us to understand a little bit more about the mechanisms of induction of immunity to malaria and other diseases. But a particular interest is that we're actually able to identify at baseline particular microRNAs which associate with susceptibility to disease. So in other words, we're able to potentially predict at baseline the individuals who are able to control the parasites or not control the parasites. And so what we're actually able to do is, for instance, I'm presenting here an example of one of the microRNAs that we've identified. And we've also shown that this actually has been already implicated in other disease models. So this particular increasing microRNA expression means worse disease. So this really has enormous potential for predicting responses to infection or vaccination. And really it can be used to develop biomarkers or diagnostics of infection, and that can be used to identify at-risk individuals or populations and used to either diagnostics vaccines designs or to monitor vaccine efficacy. And as it was just alluded to in the last talk, obviously a really practical application of this is in the field of precision medicine, and that is an area that we are really trying to move into with our work, is to sort of try and target this. So not in the context of rare diseases, but more in the context of chronic infectious diseases, which I think is an important Australian health priority area. So I hope that I've given you just a quick spectrum of sort of some of the research that we've done, and obviously all of this work has contributed many, many people throughout the world and all the study volunteers, and I would be delighted to contribute my expertise in any way I can to the Academy. And thank you again for the opportunity.
Right, have we got any questions for Denise? Steve.
Thanks, fascinating talk. I guess mine's a slightly more public healthy question, but I'm interested in whether you can answer it. There's been obviously been a lot of work on malaria vaccines, and some have shown some success. How effective do you have to make that, because it's obviously very complex and there's lots of host pathogen interactions, but is a 50% effective vaccine going to be enough? How effective does it have to be to have a public health effect?
So I think so the RTSS vaccine is based on a single protein, and that obviously has very associated with very low efficacy. And the goal is to identify a lot of to try and get around the parasite evasion. The figure that generally we're working on is about 80% effectiveness. So the RTSS vaccine is around 30% effective, and it drops off with time. So it's very, very ineffective. And part of that is again to do with the parasite adaptation. It's a big challenge, which is why we need the new technologies.
Any more questions? Ian.
It's nice work, that. Obviously, one of the problems that you have is this business of some people responding and some not to the vaccine when it's a single protein. To what extent is that due to variance in the individual, the capacity of their immune system to generate an appropriate response?
Yeah, obviously that is an important factor, which is why we sort of have the two streams. One is looking at understanding immune variation, but the approach that we're trying to do is to really take into consideration the genetic makeups of the HLA types of the individuals and try and focus on common MHC alleles which are expressed throughout the population. So with our T-cell work, for instance, we're looking at HLA supertype families, which therefore would be recognised by theoretically all ethnic, racial, genetically diverse populations. And interestingly, what we're finding, which is unexpected, is that a lot of our high priority targets are actually more conserved, which means that they're more recognised by the different strains, which is handy if that actually works out.
Any more questions? Thanks very much.
Thank you.
So thank you, Denise. So again, I'd like to thank all our speakers, fascinating talks. It's